Hello, everybody. So we're going to start very shortly. So kindly give us some time. But we'll soon start. Hello, everybody. I repeat once again, kindly give us some time. Can you hear me? Can you all reply at the chat room if you can hear me? Okay, so we are currently putting some things in place. Just give us a few minutes. We're going to start very shortly. Okay, so we're trying to set up the Zoom and the on the mic Google Meet. Okay. All right, so can you see my screen? Those of you at the Google Meet, I believe you can see the screen now. Can you? Hello. Yes, so those are Google Teams. Thank you so much for replying. So it shows you guys can see the screen. So right now we're going to be welcoming our first speaker. But before I proceed, I would like to hear from those on Zoom, right? Those are on the Zoom meeting. I would like you to reply. Can you hear me clearly? I know you can't see the screen, those on Zoom, all right? I know you guys cannot see the screen yet. Okay, so yeah, but can you hear me? Those on Zoom, can you hear me? I need you to reply with a yes. Okay, thank you so much for replying. So right now I'm going to be unmuting our first speaker while I introduce her. Okay, I'm going to be introducing our first speaker while I introduced her. Okay, so can you still hear me? At both Google Meet and Zoom, can you still hear me? I think we're experiencing network glitch. Okay, all right, thank you, thank you. And so I'll be introducing our first speaker in the house. All right. I'm trying to unmute her. So kindly exercise patience. So I'm going to be unmuting her. So why should take her up? Please, guys, can you still see? Can you guys still see the screen? Okay. Ask my hand. I'm looking for you right now. I know you cannot. Okay, I've seen you. So I'm making you co-host. Okay, I believe. Can you unmute yourself now? You should be able to speak.
Yeah. Hello, everyone.
Yes, I can hear you. So this is Ash Marhen. All right. So acromy. So she's a fish therapy scientist, in Belgium, and she's focused on the treatment of antimicrobial resistant, bacterial infections. So she has 5 years of experience in microbiology, including bacteriophage, isolation, characterization, and DNA sequencing. So she's currently doing a PhD in Belgium, and like I said, she's an expert in this area. And recently, she has also extended certain expertise dealing with genomics and certain pathogenic and probiotic, concepts of genomics. So we'll be having her while she'll be sharing from the wealth of her experience in the next 20 to 25 minutes. So we appreciate you, Asman, for joining us. So thank you. Over to you.
Thanks for this introduction. So you hear me very well, everyone here, to be sure. Okay. So let's start. As we you see my screen, guys, or so you hear me, Abraham? So do you see my screen?
Yes, we can. So please, whenever you ask a question, everybody should reply at the chat room. So just check the chat room, Asmarin.
Yeah. So okay. So let's start. So today, I will discuss with you one of the most pressing issues affecting both humans and the environment, which is antimicrobial resistance. So scientists worldwide are dedicating their efforts and time to finding solution for this problem. But unfortunately, many countries fail to recognize the severity of these issues and neglect to take appropriate action. So as you know, AMR refers to the ability of bacteria and other microorganisms to resist the effects of antimicrobial drugs.
Hello, Ashman.
Yeah.
Sorry, I'll be sending you this, I'll be sending you the Google Meet link. Kindly join with your phone on the Google Meet, okay?
Okay.
Are you with me?
Yeah, I'm with you.
Asman, are you with me?
Yeah, I'm hearing you. Do you hear me?
Yes, kindly use your phone, all right, to join the Google Meet. And don't use the video, you can just use your audio. So let's unmute yourself on Google Meet. Kindly do that so that those on Google Meet will be able to hear you clearly, all right?
Okay, so.
Apologies for that, okay? No, your phone, not your laptop.
Oh, from my phone. Okay, just wait.
Yes, your phone. So we just need your audio, okay, from your phone, okay, using my Google Meets, all right?
Okay.
So those on Google Meets.
Okay. Google Meet too.
Yes. So just your okay.
Now it's okay, yeah.
Yes, continue.
So unmute me, so you can hear me now, okay? From both.
Just continue, okay?
Yeah, okay. So antimicrobial resistance refers to the ability of bacteria and other microorganisms to resist the effects of antimicrobial drugs, rendering them ineffective in treating infections. Do you know that recently the UK government has expected concerns that AMR could result in the death of 10 million people annually by 2050. So, regarding to the problem of the rise of antimicrobial resistant bacteria, there is one contributing factor to the development of AMR bacteria, which is the presence of drug residues in animal products, in animal agriculture, to promote growth and prevent disease. However, the improper use of these drugs can lead to the presence.
Well, sorry, Asmayan, it seems they cannot still hear you.
From where? Google Meet or Zoom?
Yeah, from Google Meet. So really? I'm trying to resolve that. Sorry for all this. We had a problem with Microsoft Teams, so
okay,
we're trying to resolve.
But I think you can unmute me.
Pardon? Yeah, I'm trying to resolve. Okay, so right. Just give me a minute. Okay, so Asman, can you try unmute yourself?
It's unmute now.
Yes, unmute yourself for Google Meet, okay?
It's unmute also from my Google Meet. So
okay, go on.
I think they can hear me now. Okay. However, the improper use of these drugs can lead to the presence of residues in animal products, such as meat or milk or eggs. So consuming these products can contribute to the development of AMR bacteria in humans. Furthermore, the improper disposal of antibiotics and other pharmaceuticals can lead to environmental pollution when these substances are released into the environment through wastewater, they can contaminate soil or water and the ecosystems. This contamination can have long-term effects on environmental health, contributing to the development and spread of AMR bacteria. Also, the immunosuppression, which refers to a weakened immune system, can also be linked to the rise of AMR bacteria. When individuals have compromised immune systems, they are more susceptible to infections. In such case, antibiotics and other antimicrobial drugs are often prescribed to treat these infections. However, repeated or prolonged use of these drugs can increase the risk of developing AMR bacteria. So here is it, some of the solution to combating bacterial infections involves reducing their occurrence and developing new and alternative antimicrobial therapies. So one effective approach is the reduction of bacterial infections through various preventive measures. One of them is the vaccination, which play a vital role in preventing bacterial infection by stimulating the body's immune response to specific bacteria, by developing and administering effective vaccines that can enhance the immune system's ability to combat bacterial pathogens. In addition, botanical plants have shown promising antimicrobial properties and can be utilized in the form of plant-derived compounds or essential oils. Researching and harnessing the potential of these natural compounds lead to the development of new antimicrobial agents. Also, the use of probiotic food, which contains beneficial bacteria, can help maintain a healthy balance of microbes in the body. These good bacteria can compete with and inhibit the growth of harmful bacteria, thereby reducing the risk of infections. Incorporating probiotics into the diet can be an effective strategy to support the body's defense against bacterial pathogens. Furthermore, antimicrobial peptides, which are small proteins with potent antimicrobial properties, can directly target and kill bacteria, and making them a promising avenue for the development of novel antimicrobial therapies. Research and innovation in this field can lead to the discovery of new antimicrobial peptides or the modification of exciting ones to enhance their effectiveness. And finally, and the most important for me today, is the phage therapy that we're gonna talk about today. And it's really important as alternative to antibiotics and to fight against the antimicrobial resistant bacteria. So, but first of all, I want to give you a small introduction about the meaning and the danger of antimicrobial resistant bacteria. So AMR is a global, multidimensional phenomenon occurring in humans, animals, and environmental ecosystem. It includes the ability of microorganisms such as bacteria, viruses, fungi, and parasites to resist the effects of antimicrobial drugs that were once potent in combating infections caused by these microorganisms. AMR is a natural occurrence, arises from genetic mutations or the transfer of resistant genes between microbes. Recognizing its severity, the World Health Organization has classified AMR as one of the top 10 global public health threats confronting humanity. So, and several key factors have played a significant role in the progression and the causes of AMR. One of them is the absence of proper surveillance systems, because the effective surveillance systems are essential for monitoring the emergence and the spread of resistant microbes. Without adequate surveillance, it becomes difficult to detect and respond to AMR in a timely manner. As well, the overuse and misuse of antibiotics in both human and animal healthcare contribute to the development of antibiotic resistance. When opportunities to evolve and develop resistance, and this is one of the most cause of these infections. Also, the inadequate infection control practices in hospitals, because the poor infection control practices, such as improper hand hygiene, inadequate sterilization of medical equipment, and also lack of isolation protocols can lead to the spread of these drug-resistant infections within healthcare settings. More than that, the limited financial resources, for example, in the undeveloped countries, can prevent the implementation of effective AMR prevention and control measures. This includes investments in surveillance systems, infection control practices, research and development of new antibiotics, and public awareness campaigns. Also, the improper management of sanitation waste, because inadequate disposal of sanitation waste, particularly from healthcare facilities, contribute to the spread of drug-resistant bacteria in the environment. Because if waste is not properly treated or disposed of, resistant bacteria can enter water bodies and the food chain, affecting animal health and also human health. Also, the timeline and accurate diagnosis is crucial for appropriate antibiotic prescribing. Insufficient access to diagnostic tools such as laboratory facilities and rapid diagnostic tests can lead to empirical and unnecessary antibiotic uses, which increase the risk of AMR bacteria. And finally, the limitation of recent AMR data and comprehensive data on AMR can prevent effective decision-making and interventions. In 2019, the devastating toll of antimicrobial resistance became increasingly apparent. The WHO estimated that AMR directly contributes to 1.27 million deaths each year, making it a major public health threat. What is even more alarming in that bacterial AMR is associated with approximately 4.95 million deaths annually, surpassing the combined mortality of HIV, tuberculosis, and also malaria. The impact of vulnerable populations is particularly painful, as 1 in every 5 deaths caused by AMR occurs in children under the age of 5. To emphasize the severity, it is worth noting that just 6 priority bacterial pathogens are responsible for over 70% of these AMR-related deaths. This statistic underscore the critical need for comprehensive and coordinated efforts to combat AMR bacteria, for responsible antibiotic use, to strengthening healthcare system and research for novel antimicrobial solution. So one of the tools that can help us address these devastating damages are bacteriophages, or we can briefly call them phages, which are viruses that specifically infect and replicate within bacteria. So the history of bacteriophages dates back to the early 20th century, with significant contribution from several scientists. So the discovery of phages is credited to the work of Félix Derel, a French-Canadian microbiologist. In 1917, while studying dysentery patients in a Paris hospital, Derel observed that their stool samples contained a substance capable of killing the dysentery-causing bacteria. He called this substance bacteriophages, which means bacteria eater. Derel's groundbreaking research laid the foundation for understanding these viral entities. In the years that followed, Derel and other scientists further investigated bacteriophages and their potential application. They discovered that bacteriophages have a host-specific nature, meaning that each phage type can infect only certain types of bacteria. This specificity was a significant advantage, as it meant that phages could target and kill specific bacteria strains while leaving beneficial bacteria and human cells unharmed. So during the 1920s and 1930s, phages gained popularity as potential therapeutic agents for treating bacterial infection. Phage therapy, as it became known, involved using specific phages to combat bacterial pathogens, and phage preparation were administered to patients like either orally or topically or through injection. Some notable successes were reported, particularly in Eastern Europe, where phage therapy was extensively researched and applied. However, with the discovery of antibiotics in the mid-20th century, phage therapy took a backseat in Western medicine. Antibiotics quickly became the go-to treatment due to their broad spectrum of activity and easy to use. So the use of phage therapy declined in most parts of the world, except in regions like the former Soviet Union and Poland, where it continued to be developed and utilized. So phages are small viruses that infect and replicate within bacteria. They are generally very, very specific to a species of bacteria. Sorry. But despite their size, These phages play a vital role as the most important biological entity on Earth. They are ubiquitous in the biosphere and can be found in all environments, with a staggering number exceeding 10 to 31 on the planet. Phage contribute to the delicate balance of microbial population by their ability to kill up to 40% of all bacteria in the ocean every day, showcasing their profound impact on maintaining ecological equilibrium. So, like every microorganism have a life cycle. So phages have two life cycles. Firstly, the viruses bind to the bacterial cell and injects its genetical material in the lysogenic here. So this is the lysogenic cycle. The integration of viral genetic material into the genome of the host occurs and the bacterial cell replicate without producing variants. In the lytic cycle, viral genetic material is replicated and viral proteins are synthesized. Then an assembly of viral is achieved, followed by the lysis of the bacteria and the release of new virants. And then the bacteria is totally exploded. So the fundamental principle that makes phage versatile for several applications in their capacity to infect and kill specific pathogens and the other phage in intrinsic characteristics such as its co-evolution with its host, its enzymes capable of lysis of the bacterial cell wall, and its integration into the host genome also make them candidates for biotechnological application. That's why the application of bacteriophage is not limited to medical use in animals and the humans. For example, in agriculture, they are used to treat different problems with bacterial contamination. In the food industry, they are used as a microbiological control method for undesirable pathogens, increasing food safety. And also the proteins encoded by phages, such as indolazine, which degrade peptidoglycan, and also holins, which break the cell membrane of the infected cell, have shown important antimicrobial actions. The use of phages for treatment of bacterial infection is called phage therapy. So phage therapy is a method to combat bacterial infections using phages and has been a major focus of attention in recent years. This approach mainly uses viruses that are professionally lytic, so that they kill the pathogen without necessarily promoting any changes in its genome. Phage therapy can only be applied in the host range, is well characterized and limited to the pathogen of interest. This feature makes it possible to use phages for the treatment of infections by themselves or in combination with antibiotics, allowing the reduction of chemical use. In recent decades, the research became focused on phage as possible tools for bacterial treatment because of the rise in multi-drug resistant bacteria around the world and the decline in production and discovery of a new antibacterial chemical. So phage therapy can be delivered to humans through various methods, depending on the type of infection and the targeted location. So for example, topical application. It's used for infections on the skin and mucous membranes. Phage preparation can be applied directly to the affected area. This can involve using phage solution, gel, or spray to ensure combat between the phages and the targeted bacteria. Also, there is oral administration. Phage can be formulated into oral preparations such as liquids, capsules, or powders. This delivery method is suitable for treating intestinal infections caused by bacteria residing in the digestive system. The phages can also intravenous injection. In more severe cases or systematic infections, phage therapy can be administered intravenously. So phage solutions are directly injected in a vein, allowing them to contribute throughout the body and target bacteria present in the bloodstream or other organs, and also local injections. When dealing with localized infections, phage can be injected directly into the affected area, and this method is commonly used for treating infection in joints, wounds, and abscesses. The phage are delivered through syringe or catheter, ensuring direct contact with the bacteria. And finally, inhalation. For respiratory tract infection, phage can be delivered via inhalation. This method involves inhaling phage aerosols or nebulized solutions to reach the respiratory system where the targeted bacteria are located. So the use of phage therapy in human has shown positive effects in treating bacterial infection, particularly in the context of topical infections such as lung infections, otitis, particularly and cholera. So the most studied bacterial species in these cases include Pseudomonas aeruginosa, Staphylococcus aureus, and Klebsiella pneumonia. When employed, phages as a therapeutic tool do not distribute the normal, which are the good bacteria residing within the body. This characteristic makes them an attractive option for medicinal application. Like I told you before, phage can be used alone or in combination with antibiotics and other drugs to combat bacterial infections. During the treatment, these natural viruses multiply and increase in number autonomously, augmenting their efficacy. Additionally, phages hold the advantage of being readily available and easy to find in virus environments. Their inherent safety is noticeable, as they pose no harm to the body and are non-toxic to animals or plants and the environment, making them a promising avenue for future therapeutic advancement. Previously, it was believed that bacteriophages could not affect eukaryotic cells. However, recent studies have shown interaction between bacteriophages and various cells and systems in the human body. So phages can interact with mucous surfaces and adhere to glycan residues or mucin glycoproteins. These adherents may help shape the gastrointestinal microbiome and prevent the colonization of pathogens. Bacteriophage can also interact with cells of the immune system, influencing the expression of surfaces, protein, and pro-inflammatory cytokines. Despite the advantage advantages of phage therapy, there are some disadvantages to consider. First, one is the bacterial toxins, such as endotoxin may be released when bacteria are lysed, potentially worsening the bacterial infection. Also, foreign proteins carried by bacteriophage may include immune system reactions or imbalances in human or animals. This is also limited knowledge about phage diversity, safety issues, and proper phage selection, which need to be addressed for effective phage therapy application. Also, it's not known what dose or amount of phages should be used to treat a patient because it depends on the infection causes. As well, it's not known how long phage therapy may take to work. Until now, there not be enough kinds of phages to treat all bacterial infection, but scientists around the world are doing their best to isolate phages to infect different bacteria. Okay, I know that my presentation is very, very long, but I thought that I have 30 minutes, but it was like more than 30 minutes, I think. So just we're gonna go quickly, okay? So furthermore, phage therapy has gained attention as potential solution to address the growing threats of AMR in veterinary medicines. So like livestock, maybe the field where antibiotic use has become most abused, which has resulted in a major threat. And phage therapy has been successfully tested for the treatment of infections affecting especially cattle and swine. Also, phage therapy can contribute to improved animal health and welfare in livestock and poultry, as bacterial infections can lead to various negative impacts, including reducing growth rates, decreased feed efficiency, increased morbidity, and even mortality. So treating these infections with phages can help decrease these issues, promoting healthier animals, better productive, and improved overall welfare within the agriculture industry. Phage also been proposed as a therapy in aquaculture, for example, like the bacteria Lactococcus garfiae, a pathogen affecting several marine fish species, because there is a recent study that characterized three lytic phages against this strain, and this opens the possibility of phage-based treatment against this aquaculture pathogen. And currently, societal concern for companion animal health has increased considerably, and this has led to a misuse of antimicrobials to treat infections in companion animals, and to an increase in MDR strengths. When it comes to delivering phage therapy to animals, the route of administration depends on the site of infection and the specific needs of the animal being treated. Here are the three main routes of administration for phage therapy in animals. So the first one is oral administration. And in this method, phage are administered orally to the animal, typically throughout liquid formulation or encapsulated in food. Second one, topical administration, involves directly applying phage to the site of infection on the animal's body. This method is commonly used for localized infections, such as skin infections, wound, or infection in specific organ or body cavities. And the third one is systematic administration, which involves delivering phages throughout the animal's body to treat infections that have spread or are affecting multiple sites. Okay. So, however, the choice of administration route depends on factors such as the type and location of the infection, the animal's condition, and the specific characteristic of the phage preparation. The goal is to ensure that the phages reach the site infections in sufficient concentration to effectively combat antibiotic-resistant bacteria. And it's important to note that the selection of the appropriate administration route should be made in consultation with a veterinarian or a phage therapy specialist. They can evaluate the animal's condition, determine the most suitable route of administrations, and provide guidance on the dosage and the treatment protocol to achieve the desired therapeutic outcome. What is that? Okay, and finally, so the use of phage in agriculture and crop production is also an emerging field with the potential to revolutionize agricultural practices by harnessing the natural ability of phage to target and eliminate harmful bacteria. They can be employed in various ways to improve crop health and productivity. So the use of phage in the biocontrol of the disease management, also phage-based seed treatment can help protect seeds from bacterial pathogens during germination and early growth stages. On another hand, bacterial contamination during post-harvest handling and storage can lead to spoilage and reduce the shelf life of agricultural produce. And also for the soil health and phytobiome management. Okay. Okay, so we don't need this disadvantage. So our conclusion. So to summarize, the use of phage therapy in different fields has the potential to benefit the environment and public health by reducing the reliance on antibiotics. Phage therapy helps mitigate the environmental impact associated with antibiotic usage and additionally minimize antibiotic-resistant transmission, and also the environmental and public health benefits of phage therapy align with the One Health approach, which recognizes the interconnectedness of human health and animal and environment. And finally, there is complementary strategies. Is important to know that phage therapy is not meant to replace all antibiotic usage. It's considered a complementary strategy that can be integrated with other measures such as good hygiene practices, vaccination, and prudent antibiotic use. And really thank you for your attention, all of you. And sorry to be late. I don't know if you have any question, I can answer it, of course. So Abraham, you can. Hey. All right. Thanks for you. I'm really sorry to be late.
Yeah, thank you so much for joining us this time.
Thanks.
So we really appreciate you for joining us.
Thanks.
I hope I'm audible.
Yeah.
Okay, thank you so much. So we'll be having our next speaker, but before that, do we have any question quickly? Do we have any question in the chat room?
Quickly. There is one from Google Meet. I don't know. I can't hear her. So, like, could you like just type it? How will we get the certificate, please?
Okay, you guys will definitely get your certificate by the end of this program, but only for those who wait. So we're going to share it on the chat room so that you guys can get your certificate. Okay. So meanwhile, yeah. So that's it basically. Thank you. So, but based on the presentation we have, do we have any question?
Is there not any bacteria phage that is
okay? We
pathogen?
can only take one or two.
No phages. They are not harmful for environment or animal or human. So they are like they just targeted the pathogen bacteria, not the good bacteria. So they are not. But there is like. There is some toxins in some phages, and we can like just extract them from these phages, and we use the phage to treat bacteria.
Amazing.
So for Arogandin, I don't know.
Okay, so do we have one more question? Yeah, please go on. Thank you.
Thank you very much, Doctor, for this wonderful presentation. Please, we are a little bit confused that for clarification is when you're talking about the administration of this phage.
Yeah.
I made mention of our administration, and also going back to the disadvantages of this phage. You made mention of production of
toxicants. Yeah.
So if the disadvantage is the production of endotoxin, then why would it disturb oral administration? Thank you. I just need more clarification on that. Thank you, Doctor.
So thanks for your question, but do you mean like if there is on the toxins in the phages, so when we can use the phage? Like for which?
The question is if it's developing toxin, why should we then use phage? I think that's a simple.
We can use even if even the phages that they have toxins, so we can just like eliminate this on the toxins by some protocols, okay? And after that, we can use these phages.
Okay.
Like for oral, for example, administration or topical.
All right, thank you.
Yeah.
So I hope it's clear. So probably, can you share your slide with everybody? I think they're asking for your slide. Is that okay, Doctor?
Yeah, I can send it to you. Okay.
All right. All right.
Yeah,
so
sure. I'll send it to you.
Okay.
Yeah.
Thank you so much, Asman, for joining us. We really appreciate it. Yeah, looking forward to having you next time.
Yeah, sure. Thank you.
All right. So please, can we appreciate the chat room? Everybody, please drop appreciation, but I will thank you at the chat room to appreciate our first speaker.
Yeah.
So while we're doing that, we'll be having the second speaker, who is the person of Mr. Malik, and he'll be taking us on the workshop, all right, on transcriptomics. So he'll be talking to us about transcriptomics, and he'll be showing us some brief hands-on training, okay, on transcriptomics. So I believe Mr. Malik is here. So after the workshop, we're going to be talking about DNA sequencing and its application in life sciences. So we're going to be having Dr. Faladi from the US, from the United States. So she will be presenting on the DNA sequencing and its application for like some minutes for us before we round up. So at the end of this program, we're going to be providing the link for you to get access to your certificates for 4 months in a webinar series on genomics, bioinformatics, and career opportunities. So, yeah, thank you. So, Dr. Malik, are you here with us?
Yes, hello, I'm here. Can you hear me?
Oh, yes, we can hear you at the Zoom, but we cannot hear you on Google Meet. Please, somebody, can you mute yourself? We'd really appreciate
Yeah, just
that.
a second. Let me switch to
Yeah,
mute as
unmute
well.
yourself for Google Meet, okay? So that we can hear you.
Yeah, sure.
Have you done that?
Yeah, I just put a request. If you could add me.
Okay. Yeah, now you can do that now.
Yeah. Oh.
So I would advise you use your phone.
Yeah.
Okay.
So, okay,
okay,
so
you
can
can go
everybody
on
hear
now.
me?
Yes, we can hear you. Go on now. Thank you.
Okay. Yeah, let me share my screen. Yeah. So I hope everybody can listen. Okay, my name is Hafiz Muhammad Talha Malik. Just a brief introduction of myself. I've done my master's in bioinformatics and currently working as a senior bioinformatician at ABO Scientific. Well, I've got almost 7 years of experience in the domain of bioinformatics, and I've been working mainly on the in the domain of bioinformatics analysis and mainly in the omics and data analysis. And I got few publications as well, which are mainly related to the omics analysis. What I will be covering today is towards the RNA-seq, and I will be mainly focusing on the biological aspects, bioinformatics analysis aspects of RNA-seq. So I think as the title of this webinar suggests that, and as Dr. told me that we have a series of webinars in this domain, genomics and bioinformatics. So I presume that most of you must have known about this RNA-seq before as well. And obviously, you must have gone through what's the basic molecular biology of it. So I'll try to be more concise and towards the bioinformatics side of it. Just a brief introduction to the RNA-seq. So basically, RNA-seq is a high-throughput sequencing technique that allows us to measure the expression levels of all the genes in a sample. And basically, what we do is we take the DNA and convert it to cDNA, and then go towards the sequencing, and then the data comes from the sequencing, and then we go towards the bioinformatics analysis of it. There are many uses of RNA-seq, and it's not only limited to the RNA-seq. We have now single-cell RNA-seq and also the snRNA-seq, which is single nucleus RNA sequencing as well. So, but today my demo and my presentation will be more focused towards the whole RNA or mRNA-seq analysis. What are the main uses, you can say, of RNA-seq? There are multiple uses, and what we can do is we can analyze the gene expression analysis, which says that how the different conditions or biological conditions have different level of genes expressed in them. So we can compare between different conditions and how their expression goes out. Then the transcriptome profiling, you can profile the individual's transcriptome. You can also detect the non-coding mRNAs, non-coding RNAs, and long non-coding RNAs and circular RNAs as well from the RNA-seq data. Then you can do the functional annotation where you can annotate the differentially expressed genes or whatever genes you have identified from the RNA-seq data to from the database which have already the published data or they have the information about those genes. You can do that. Then also you can do the pathway and enrichment analysis where you can check which pathways are differentially expressed or which pathways are playing part in the genes which are being differentially expressed. Then you can use RNA-seq analysis and the splicing. You can like study the alternate splicing events, and these basically contribute towards the transcriptome diversity as well. And also fusion gene detection, where you can detect the fusion genes and basically which are formed by the rearrangements, and these can play and these do play an important role in the cancer development. One question may arise that why we are using RNA-seq when we have the microarray. Well, there are a few advantages of the RNA-seq over microarray. As one is that microarray is quite an old technique, although people still use it, and also people use it to verify the RNA-seq analysis as well. But there are mainly a few very good advantages of RNA-seq over microarray. One is that it can detect lowly expressed genes, and also the other advantage is that RNA-seq gives you the whole picture or whole transcriptome of very individual or a sample, whatever you call it. And in contrast that you, when you use a microarray, you have a limited probes and coverage, which can, which leads to the missing of important transcripts or regions of the genome. Also, RNA-seq helps you in detecting the novel transcripts, and as I mentioned, you can detect the alternate with splicing events and fusion analysis as well. And also, one of the main advantages that it is more accurate and reproducible. Well, one advantage is although the price is still higher, but as the things going on, we can say that price is coming down a bit and quite heavily, like it's coming very heavily. In case of RNA-seq analysis, the one major step is the designing of the right experiment. You have to design your experiment very precisely and very rightly. The main question is how many numbers of samples do you need in the RNA-seq experiment, especially when you are doing the differential gene expression. So as we go through along this webinar, we'll see that there is a lot of stats involved in this RNA-seq analysis. But to be very concise and very short, I can say that as we are measuring the counts or measuring the gene expression in terms of the gene counts or read counts, whatever you call it, the minimum number people suggest is for the one condition should be 3. Although some people say that 2, you can go with the 2, but mainly people suggest that you have to be, the number should be the minimum of 3. So you have to have 3 minimum samples from each condition, and then you can compare with them. Then again, the coverage is like how many number of reads you are sequencing and how the coverage depth you are taking to cover each transcript. And then the read length, either the read length should be short or long, but that long or short, but that depends on your experimental question. And again, long read has its own advantages and disadvantages, and long short read has its own. But to be very short, I can say that short read has very much accuracy, but the long read does give you more giver picture, and they can give you more accurate results in terms of you can cover the more area, and there are the chances that you will miss less area. That's in a short and a bit, but again, the short reads are less expensive, and they are more accurate, but the long reads are still expensive, and they are again not much accurate, they are more error prone. So just library preparation is the first step in the RNA-seq experiment, where you take DNA from the samples, and you convert it to cDNA, and then all those library steps where you do PCR, and you attach adapters, and then you go to the um, sequencing side of it. Once you are done with the sequencing, we comes up with the data. Your data can be in the form of a single end sequencing, where you have one um, file for the each sample, and then there are a paired end sequencing, where you have two files for each sample. I hope everyone does know about the FastQ, or where the data when the data comes, how it looks like. I'm not going to go into the details, but I will try to go through again the analysis aspect. So once we have the data, we have sequenced the RNA-seq analysis, RNA samples, and now we have the data in the form of FastQ files in our computer. So what what do we do? So there are quite few pipelines you can follow, but this one I'm showing you is the most common one, or say mostly used one, where you get the data, you clean it, then you map it to the reference genome, then you count those transcripts abundance, and then you go towards the differential expression, that how many genes are upregulated in one condition and down in another, and vice versa. So basically you are comparing the two conditions, like I say one is a cancer, any cancer, and the other is normal patient. So you are comparing the gene expressions of those two patients, like one is the normal one and one is the cancer patient. So you are basically comparing the gene expression, which genes are expressed in one condition, and which are expressed more in the other condition. That's what a differential gene expression pipeline looks like. So the basic step is the quality control of the data. You have to check the quality of the data, where you check if your data is coming from the data which comes from the sequencer is of good quality to go through. So once you have to make sure the data is good enough to proceed. Once you are sure that your data is good enough, then you can proceed to the next step, because the quality of data is very much linked towards the end results of the analysis. So your data has to be of good quality, and there are multiple tools you can do that. The picture you are seeing is from the FastQC tool. You can give your FastQ files to this tool, and it can give you the basic statistics of your data, how its data looks like. And if you can see from the picture, there are multiple parameters on which the data is being measured, or data is being assessed. The green one says that it's a good quality. These measures are being good. Red means like there is a problem, and yellow means like there is a problem. You can proceed with it, but you can do you can clean the data as well. So again, I'm not going towards its details, but just to be specific, first step is the quality control. You have to check the data quality. Once you're done, you can go towards the alignment. So alignment is, you have the raw reads, your short reads, your RNA reads. You are going to map it to the reference genome. It can be the genome, it can be the transcriptome. So if you map it to the genome, you will get more probably towards the differentially expressed genes. And if you map it to the transcriptome, you're gonna get the differentially expressed transcriptomes. So that's your choice, whatever you want to use and how you want to proceed. There are multiple tools available for this. Again, these are tools which are being widely used. HiSet2 and STAR. These are very common and popular these days for the RNA-seq data alignment. So what you do is you map your data to the transcript, your reference genome or transcriptome, whatever you are using. The next step is the quantification. Once you have done the annotation. Oh, sorry. Once you are done with the mapping, you have to count that how many reads are mapped to one gene, how many reads are mapped to the other gene, and this goes for the multiple samples as well. Like for one sample you map, you will check that how many genes, how many reads are mapped to the gene 1, gene 2, gene 3, gene 4, and then same goes for the all the samples. You have, like, if you have 6 samples, you go to the same stem for the 6 samples. And there are, I'll show you this sample file as well, but you will get from that the quantification step. And there are many tools for this step as well. One of the more popular is feature counts and HTSeq. So once you are done with these steps, then comes the differential expression analysis. So again, this is a lot of stats involved in these steps. I'm just gonna go briefly how we measure the differential expression from the RNA-seq data. So basically what we do is we use the statistical testing to decide whether there is a difference in 2 different conditions or not. So we are checking, like, if we have 2 conditions. So which condition has genes upregulated and which has the downregulated, and vice versa, that which genes are being upregulated in one condition. So obviously they are being down-regulated in the other. And if one gene is down-regulated in one condition, then it means that it is upregulated in the other condition. How we do it? So again, there are quite a lot of stats involved in it. I can add it. Yeah. So basically what we do is we have like 2 genes. Like this is one gene and this is gene number 2. Can you see the editing, right? Or the pen I'm using? Yeah. Okay. Yeah, if you could just tell me, like, you can see the screen that I'm what I'm doing on the screen. Yeah. Okay. Okay. So we have 2 genes. This is gene 1 and this is gene 2. And this gene is of, I say, 1000 base pairs, and this gene is of 500 base pair. I'm just using an example. So I say there are 2 reads mapped to this one and 2 reads mapped to this one. So if someone could just comment, like, what do you suggest, that is these 2 have the same expression, or they have a different expression? What do you suggest? Just if someone could comment on the Meet. I'm using Meet on the mobile, so I can see those messages on the Meet, not on the Zoom right now. Yeah, if someone could comment, just random thoughts. So which gene do you think is more expressed? The first one with the 500 base pair and the other one with a 1000 base pair? Like, no comments? Or so one is saying the 1000 base pair is different, more expressed, and 501. Yeah. So what I can say is, from the look of it, you can say that the 501 is more expressed because there's length factor as well. Like the gene is short and the 1001 is less expressed because we can assume that not the whole gene is covered, or we don't have enough confidence to say that if it is more expressed or not. But again, there's a lot of stats involved in this, that I'm just showing you idea how we're going to do it. So if I say this, I'm only not looking into this number of reads mapped to this gene, but I'm also looking at how many number of reads mapped to the each sample, how many, like millions of reads are mapped. So I'm going to take account of that as well. And also I'm gonna take the measure of the variance as well, how and the mean as
Yes,
well,
we
how
cannot see your
each.
screen.
Oh, you cannot see my screen. Okay, let me try again. Yeah. Okay, can you see it now?
Yes.
So gene expression, we have to account for multiple things. One is the gene length. Then one is the how many number of reads mapped to that each sample. Then we have to account for the um, variance as well, and the means as well. And when we go towards the testing as well, which is kind of a t-test, not exactly the t-test. It's a kind of a t-test where we check the means of those samples based on these measures, and then we come to the conclusion that which genes is upregulated and which one is the downregulated. For the whatever I've told you, we account for these steps. We basically called it as a normalization step, where you account for those things. Basically, we have to normalize the data so that we can be sure that each sample is or each gene or each sample from each condition is being taken equally. And this also includes the batch effect, where if you have um, samples coming from two different runs of sequencing, then you have to account for that too. Basically, this is called the batch effect correction and also the normalization. We'll just go towards how we do it as well in the demo part. I'll go through multiple steps how we do it. So the normalization, when we do normalization, we have different measures of it. We can go for the RPKM, FPKM, TPM, and most of you must have heard about it. So I'm not going to go into the details of those, but just to let you know that these are the normalization techniques where you account for all these things from your data, and then you go towards the differential expression. What are the different tools used in this? In the differential expression, the whole pipeline, I am sorry, seeing you there. If you have a reference transcriptome or genome, you can use top hat, bow tie, HiSet, star. There are many tools. And then for the differential expression, you have different libraries in Millen or Edjar or Limar, the mostly used. And then there are many others and still coming up, where each of them are basically different from other technique based on how they calculate these normalization steps. What kind of plots do you can make? And from this analysis, you can have bar plot, you can have MA plot, you can plot the PCA, where you can check how many or these how many samples are grouped together and how samples are grouped together. If you have two conditions, so this will give you an idea that if there are two groups, or all those samples belong to group one are grouped together, or there is some difference in them. Then you have heat maps. You can plot different pathways in R, and you can also do the volcano plot, where you can plot all the differentially expressed genes. On the left side, you have the downregulated genes, and on this one, you have upregulated genes. It's RNA-seq is not limited to differential expression only. We are like going in the era of AI and machine learning. So and it is being widely used nowadays. Everyone is fascinated about how AI is being used in RNA-seq and genomics, and we all are being fascinated with these terms. So RNA-seq is you can use AI or machine learning in RNA into the biomarker discovery. You can subtypes or classify the disease based on their expression. You can use a drug response prediction where you can detect how individual um, are likely to respond to the specific drug. Again, then you can identify the therapeutic targets using the AI and machine learning techniques. And again, you can monitor the treatment response, and you can track the transcriptomic changes. And also we can go towards the personalized or precision medicine and personalized cancer immunotherapy as well, where you can detect based on one's transcriptome that which kind of drug is suitable for that kind of a person. This is what we call as a precision medicine or personalized medicine, and you can also use in the immunotherapy, cancer immunotherapy as well. That's it from the presentation side. If you have any question related to this presentation, you can ask, then we can go towards the short demo of how we do it. If anyone has a question, he can or she can ask. Any questions? I think someone has raised his hand. You can ask question. Yes, please, you can type your question here. Why did you choose this topic specifically? Like what topic? Differential gene expression? That's Larry asked this question. Why did you choose this topic specifically? Are you asking me like why I choose this differential gene expression? Okay, there is another question why it is necessary to convert the DNA into cDNA first, because currently we don't have the technology where we can directly sequence the RNA. So what we do is we convert it to cDNA and then we sequence it. That's the question from Muniba. Yeah, sorry, Mariam. Right, yeah, why did I choose this? So basically, the idea was to just to let you know about the transcriptome analysis, and so I'm just going through the one of the techniques or one of the pipeline we can do from the RNA-seq. There are multiple we can do, and I think when we will have a workshop on the transcriptome analysis, we can discuss the further in the details as well. Extra sequences, and how can we remove the extra sequences like primer sequences? Okay, so you can use different tools like Chemomatic, and there are many tools after QC, FastP, CutAdapt. You can trim your data from those tools. That's the question from, I don't see your name. Adnan. Yeah, that's what you, MegaX, no, MegaX is not used. I don't think so. MegaX still has anything related to RNA-seq or NGS adapter trimming. We don't have anything right now. There are a few other questions. Please, how do we query database to get data we actually want? For the RNA-seq, I don't quite read the name exactly. Shimoric on sorry, I can't read your name. How can like if you want to get the data, download the data, you have NCBI, you have Gene Expression Omnibus database, and there are many cancer for the cancer. There is a Cancer Atlas database as well. And What are the approaches for expression analysis? Sorry, can we do RNA-seq analysis through CLC genomics? I think so. We can do that using CLC genomics as well. What are the approaches for different for expression analysis? What expression analysis approaches? Well, there are different approaches. You can use a reference guided approach where you map to the gene, map gene to the reference. The other alternative approach is de novo, where you can assemble the transcriptome and then you go towards the expression, differential expression. Okay, 31. 31 is slide number 31. This one. These are different plots. These are basically clustering based plots. One of them is a PCA where you can see the two different groups coming out of it. Basically, PCA is a technique to cluster your data. So like I can say there are 1, 2, 3, 4, 5, 6, 7, 8, like 10 samples in the group 2 and almost 10 in the group 1 based on the different colors. So you can basically cluster your data, like if your data is from all those samples which belong to the group 1, are they coming together, are they are together in a sense, are they based on read counts, and or those are belonging to similar condition or not, and or those their variances are like closed together. That's how I can define it. Okay. Yeah, I'll share the presentation. Which programming language is best for this? Mainly we prefer R for this. Sorry, I didn't explain the second plot. Basically, the second plot is a differentially expressed genes. And it is basically a heat map for genes. And the rows we have the genes, and on the columns we have the samples. So which gene has expression high in which sample, and which has a low. And based on color, you can differentiate between them. What is the difference between DG analysis using transcriptome and reference genome? Okay, for the transcriptome, basically there is main differences when for the transcriptome we have basically the transcriptome or the whole transcriptome of the data. In the genome we have the whole genome. So in the transcriptome you are only with the mRNA and transcripts of every gene. You won't have the non-coding part in that. So when we do it using the transcriptome, we will get differentially expressed transcripts, and for that genome will get differentially expressed genes. Okay, let's go towards the analysis part. Just to show you sorry. So basically, when we, I'm just skipping the mapping and the quantification part, just because I know, I think you must have gone through this. There are, I presume that you have gone through the genome and microbial assembly as well, or microbial mapping as well, where you can have a reference genome, and the raw reads you can map it using BWA, you can use bowtie, you can use HiSat and star, whichever tool you prefer, you can use it. When you use when you do the mapping, then you do the quantification. In the quantification, in the detailed workshop, I'll be going through those steps as well, but I'm just going right now for the mainly towards the R part, where we get the count data. So basically count data looks like this. You have this gene. Basically, this is the test data from Arabidopsis thaliana. These are the genes, and these are the read counts for each sample. This is sample 1, 2, 3 for the mutant group, and these 3 are from the control group. So basically, what we do is we count the reads in each sample and each gene. So what I say is this is a gene number 1, and it says the mutant or the sample 1, there are 544 genes mapped to this gene in this sample, and 668 in this sample, 231 in this one. And then for the other group, the sample 1 has 538, 618, 714 in each sample. So then we go towards the differential expression of it. We count all those measures where you go account um, other size factors and other things, and then you will get to know those if this gene is differentially expressed or not. Right? What we do is we take this file and we import it in R. So R is basically a programming language where you can basically, it's a statistical scripting language where you can do the statistical analysis of your data. You can do whatever type of statistical analysis you want to do. There are multiple libraries for, and millions of libraries for doing the differential all kind of statistical analysis. For differential gene expression, we have some specific libraries like DEC2. So I'm just gonna show you, can you see my R screen? Yeah. Yeah, so this is basically R Studio. I have a script written in it, and this is my console where I can do or whatever I want to do. I can do 1 + 1, and it will give me 1 + 1 is equals to 2. So basically, R is built for statistical analysis. You can perform many kind of statistical analysis on it. So I have this data. I just showed you. You can't see the screen. Okay, let me share it again. Okay, can you see the screen now? I think so. Yeah. Okay, so this is the data which I just showed you. Showed you we have these genes here and the number of reads mapping to it, and we can just simply import it in R. Let me just set the path, and then I have this file. It is a CSV file. RNA-seq data is the name of it. So and basically these are the libraries I'm using. Library is basically a defined function which knows how to process the data if data is in a specific format which is written back in the backend. So if data is in that specific format which this library needs, it can perform specific function on it. I can give you a simple example, like if we use calculators for the sum function, like we type 1, then press 1, and then plus 2. So 1 + 2 is equal, it will give me an answer of 3. So how that calculator knows that 1 + 2 is equals to 3? So basically the function is written on the back end that if whenever there is a plus sign coming to it, so you have to add those numbers. So this is basically a function. And these libraries are basically written in a way that you can use that function, and then you can use those libraries to perform whatever analysis you want to perform. So in this step, I'm using this DSeq2 library. I'm just going to load it. Then there's a ggplot library. Basically, ggplot is for the plotting, different kind of plots. You have seen a few plots in my presentation as well. You can use that library for it. And then p heatmap library, you can use this library for the heat map functions. So this is a function. I have written this code. What I'm doing is I am reading this file into this variable count data, and this is my file which I just showed you, this Excel file, and I can use the read.csv function to let the R node that my file is in CSV format. CSV is basically comma separated file. So I'm just going to read this function. Sorry, type this code and this will read the data. So you can see that I have this file coming in this R studio or R language. And I can view my data in this here as well. So where you can see these are my gene IDs and these are my samples where all those counts are coming from. In an RNA-seq experiment, or when you do the differential gene expression analysis, you have to define your experimental design to the computer. So the experimental design is, you know, that these first three columns belong to the mutant group, and my next three columns belong to the control group. I know that, your lab fellows know that, your professor knows that, but computer doesn't know that. You have to tell the computer that this is my experimental design, that these samples belong to this group and these samples belong to this group. So in this code, line of code, what I am doing is I am explaining the computer or this programming language that I'm basically explaining my experimental design to it. So what I am saying is my first three sample belongs to the mutant group, and my next three samples belong to the control group. This is how computer will get to know that these samples are belong to which group. And if I simply view this, so it is saying that first sample is mutant, second is mutant, third is mutant, and the rest of the three are from the control group. And basically we have two levels from the control. We have two levels, control and mutants. The next step is, as I told you, that we have different libraries or packages in R for the differential gene expression. One of them is DE-seq. So what does DE-seq do is it does the normalization step, and it will give you the differential gene expression. So for giving you the data, the R language data we have to give the data in a specific format. And this format is defined as DE-seq set format matrix, where you have to give the count data, where this matrix, you will give this matrix. Then you have to define these columns. We have a number of samples columns, and it belongs to this condition. Like what I am telling to this object is, I have this sample, these samples, and they belong to this group. And basically I have two conditions, control and mutant. So when it will perform the analysis, what it will do is it compares the controls versus mutants, or mutants versus control. You can set how you want to do it, control versus mutants or mutant versus control. So basically this is important because you need to know how you have defined your experiment. Because if you define as control versus mutant, then the upregulated genes will be when it will give you the stats of those genes. So those which are upregulated, you got to know that the upregulated are basically in the control group, and downregulated are in the control group. And but when you do the otherwise, the experiment will reverse, like those genes which are expressed in upregulated in the control group, they are downregulated in the other one. So you should know that which condition you are basically setting as a base condition, and the analysis or the results you are getting. So you get to know that if it is upregulated, so in which condition it is upregulated. That's a very important step in this. So what I'm going to do is I'm just defining this object, this DDS object, and then I am performing the DE-seq analysis. This short code will do the differential gene expression. So what it is doing is it is estimating the size
Hello,
factor.
Mr. Mali.
Yeah.
So we are almost out of time. So
Yeah, but just give me five more minutes. Five more minutes. Okay, so yeah, this basically what this code did is it perform all these steps. It's size vector. This is counted the dispersion, dispersion relationships, and then it finally fit the model and do the differential expression. And we can get those results in my result variable. What it will do is you will get those all those stats where you have gene ID, then it's a base mean, log to fold chain. Log to fold chain means based on fold chain, you will get to know that which gene is upregulated and downregulated. Then you have a p-value and the p-adjusted value. P-adjusted value is another um, a, a method to basically correct the p-values. And what I can get the summary of it as well. So I get to know that how many of genes are upregulated and how many of them are downregulated. So my data was basically a very short data, and it has total of 100 genes. So and in your data you will be having like thousands of genes. So this summary tells me that there are one gene which is upregulated because it has the um, log fold change greater than 0, and there are 5 genes which are downregulated. And I can simply subset them in this as well, and we can plot different plots as well. We saw those. I'm just going to show go through these codes and let you know. This is the one of the bar plots where you are showing the library sizes and how many number of reads are mapping to um, which sample. So basically which we call as the library sizes. Then we have a PCA plot. PCA plot, basically you can group your data and you can see how each sample is grouped. These are the mutant groups and these are the control groups. Although they are quite spread, but in an ideal condition, they are very much grouped and close together. Then you can perform the heat map. Where you can plot number of different, basically what I did is I differentially expressed genes. I plotted those differentially expressed genes. And another one is the volcano plot. You can basically do plot your data in the kind of volcano where these red ones are the upregulated and these green one is basically, sorry, this green was is downregulated, and these red ones are downregulated. That's a small glimpse of what RNA sequence analysis looks like. So if you have any question, that's it from my side. If you have any question from the audience, I can answer them.
I think we have a question.
Yeah, sure. Please tell us application of detection micro. Yes, you can do the microRNA as well, and more of the analysis will be look like the same, but the library preparation step will be different, and you can do the microRNA analysis as well, and you can detect them though. Any other question? Okay, someone which non-coding, so okay, your question is, you said we can use RNA seq to study the non-coding. Yes, we can detect RNA, non-coding RNAs as well. There's a whole different pipeline for this thing. Yes, and we can do that. So in the short, yes, we can do that, but there is a whole different pipeline to it, where you can do the assembly and then you detect those non-coding RNAs. It's a different pipeline. I think it's a quite long question. Again, one of those steps involve are you do the transcript assembly using Top Hat or whatever tool you like, and then you use the reference annotations where you use the reference annotation and you map your assembly to it So you can get to know which data or which reads are mapped towards the non-coding part, and then you can extract those. Yeah, you can have my email address. I've shared in the presentation, and when I will share my presentation, you will have that.
Yeah, as for the presentation, I think you can share with us. So share with them.
I'll share with you, and you can share with the rest of them.
Yeah.
Any other question?
Okay, I think that's okay for today. Like the questions, so we'll be seeing you. I think, do you answer that of Google? Google Meet. Is there a tool that can detect and delete rRNA from whole genome RNA set data?
Is there a tool that will detect and delete rRNA from whole genome? I haven't seen any specific data, but what you can do is there is a very specific type of thing. But what you can do is you can map your data to the transcriptome, where you will have only the mRNAs, and then you can extract the only mRNAs, and you won't be having rRNA data in it. That's it. Is that it? Yeah. I think that's it from you. Thank you. If you don't have any question, I'll share.
All right. Thank you. So we'll get a presentation I will share with you guys, the group.
Yes,
so meanwhile, we have something else to share with you guys. So still based on the transcriptomics analysis. So because I believe you guys are, really exposed to what transcriptomics data analysis is, and the practical applications of this in different fields, such that it is applicable in microbial, it is applicable in plant, and it's applicable in animal research. And to be a very powerful tool in your hands as a life scientist, all right, to be able to use this in your various field of study. And which is why I'm going to be presenting sharing my screen right now. Okay, I'm going to be sharing my screen right now. I hope you can see my screen. I hope you can see my screen. I need a reply. I need a reply quickly. Can you see my screen with a yes? Okay, so we're presenting RNA-seq because we don't really have a lot of time, like much time to really show you guys deeply the practical applications of of this. So and it's a whole lot. So we are presenting an RNA-seq workshop. All right, an RNA-seq workshop. So and it's going to run for a very good 5 days. All right, it's going to run for a very good 5 days. So day 1, day 2, day 3, day 4, and day 5. So as you can see here, we have the lecture, which covers the overview of RNA-seq in detail. Then we have working on, assessing RNA-seq data from available databases. And then we have hands-on Linux. So we're going to be introducing you to Linux command line, the basics that you need, or some of the major fundamentals experience you need in exploring RNA-seq data analysis in Linux command line interface. And then we're going to introduce you to FastQC and cleaning and assessment processes. Then we're going to introduce you to hands-on alignment and mapping, okay, of the RNA-seq data to a known reference or whatever method we're going to be using, the transcriptomics approach, okay. And this is going to be done through some genomics, transcriptomics tools, bioinformatics tools, which of course you're going to, because this is going to be hands-on, all right? It's going to be entirely hands-on. And then we have some kind of introduction to R. So we're going to take you through R, and then we're going to take you through practical process and using R to carry out differential gene expression analysis, and then have some visualizations and plots, and also explore the pathway analysis of the RNA-seq data, from the differential gene expressions, in order to understand the approach in this. So basically what we're looking at is basically the tools, the skills or the tools that we're going to be exploring include Linux, okay, include R and transcriptomes, or let's say bioinformatics tools, okay, certain bioinformatics tools, advanced bioinformatics tools used, all right. So we're going to be looking at this basically, okay. So these are the major things that we're going to be looking at. So now, actually, the choice is yours. The cost for this, workshop for 5 days is like $150. So, but then we're giving you a kind of discount, and the bonus price we have right now is $30. All right. So for those who will be interested in this training, all right, for those who be interested in this workshop, we don't necessarily need a lot of people. You get my point? So, but if you know that this is valuable for you, and then you really want to explore or acquire the transcriptome data analysis aspect of this of bioinformatics, so this is like a very good opportunity for you to explore. So this is the time for you to to subscribe or join the hands-on training on transcriptome data analysis for 5 good days. So we have the day 1, the 2, the 3, the 4, and the day 5, and it's going to be starting by next week. So we're going to let you know the date. So I would like to know if you're interested. Can you reply with a yes or reply with I am interested? Hello. If you're interested, please can you reply quickly? Can you reply with a yes if you are interested in this workshop? I'm waiting. So probably there could be more discounts. It's possible that we have more discounts, but we need to know how many people are willing to take the workshop or to partake in the workshop. So thank you. I'm seeing a reply at the Google Teams. Okay. All right. So it's going to be virtual. Okay. Yeah, 1 hour per day. It's going to be virtual. Okay. And it is going to be practically explored. Okay. So we're going to be doing this from the basic to the advanced. We're going to be doing it on Zoom. So like I said, we don't necessarily need many people. So, but we only need very serious people who would like to explore this area of study. Okay, so we're going to be sharing the link on the WhatsApp group for those who are interested. And like I said, it's going to be $30 instead of $150. And like I said, our course instructor is the person that just finished presenting. All right, and he has like 7 years of experience in working with clients, with researchers, with institute like companies and things like that. So it's going to be taking you through the process and share with you, his experience in a very deeply way. Okay, so Kareem Abdul, yeah, I assure you that it's going to be very deep. Okay, I assure you that it's going to be very, very deep. So you just need to enroll, and then you see most of our courses. You can ask for those who have enrolled in our courses before. It has always been very, very deep. So, but I will discuss with the course instructor if we can actually advance the time forward so that you guys could like have some time to be able to talk with the course instructor and have some kind of experience with him. Okay, so like some kind of like maybe we can have more time to the time that we have. So this is the learning objective. In this transcriptomes workshop, you learn the objective, which will include how RNA-seq technologies are applied to microbial, plant, and animal research. Understand, this, and then to learn the importance of FastQC, develop Linus, skills specifically relevant to the animal, plant, and microbial transcriptomics. Gain practical experience in quality assessment and clean transcriptomic data for these organisms. Master alignment and mapping tools, quality, of gene expressions analysis, the use of RNA-seq for the visualizations, pathway analysis, engage in question and answer sessions, and probably we'll have some time more to be able to explore this. So if you are interested, we're going to be sharing the link on the WhatsApp group, the link of the group on the WhatsApp group so that you'll be able to join the program. Okay, so that you'll be able to join the program. Meanwhile, we have the certificate. We have certificate for the 4 months, and we want to congratulate everybody. It's really been a long time. So, but then we have one more person who will be presenting between 15 to 20 minutes. Okay, presenting to us between 15 to 20 minutes. So she's a doctor from the United States of America. Okay, so she'll be presenting to us on DNA sequencing and its applications in life sciences. So wait behind while we are working on the certificates to share the link with you guys. Okay, so I don't know, is she Dr. Faladi? Are you here with us? Hello? Hello, ma. Are you here with us? Please, those on Google Meet, can you hear my voice? Can you hear me, please? Please, can you hear me? Can you hear me now? Hello, can you hear me now? Okay, okay. So we'll be having Dr. Falade to present to us while we work on the certificates. So so I'm speaking to her so that she can join us on the call while we will be sharing the link. Okay, we'll be sharing the link for you to join the Transcriptomics workshop. And we'll also be sharing the course outline. Okay, we'll also be sharing the course outline. So hello, Dr. Falade, are you with us? Hello, Dr. Falade. Okay, while she's preparing to join us, let me quickly share my screen. So we have one more thing that we would like to share with you. So in case you're interested in a full-scale research on comparative analysis and functional metagenomics, so this particular program will run for 3 months. Okay, we run for 3 months, and it's going to be from research project to published paper. All right, and then you, at the end of the day, you have a research paper. We work on full-scale comparative analysis of metagenomic sample from soil, from water, or from whatever environmental samples you want to explore. And then at the end of the day, you're going to be having a research paper. So this is a full-scale course outline that we're going to be having. So we're going to share the links on the group as well, if you, in case you're interested to join. The actual cost is like $800, but we're going to be giving out at $100. Okay, 3 months full-scale research, $100, and then you're going to be having a research paper that you can publish on your own at the end of the 3 months. So it's going to be full-scale research, hands-on training, practical guidance. It's like internship, and then we're going to guide you through to projects, do a lot of stuff. If you don't have a personal sample, we'll take you through online database to get a kind of samples that will fit into your biological questions. So if you're interested in this, I would like to see if anybody is interested in the metagenomics research. Anybody, please? So I'm waiting. Do we see anybody who is interested in the metagenomics, please? Metagenomics, okay? In the metagenomics. So Dr. Falade, are you with us? Hello, ma.
Yeah, I had to come back.
Oh, okay. Have you joined, ma?
Yes, I've been online for a while.
Okay, ma. So what's the Zoom name, ma? The Zoom. Sorry, I'm looking for her so that I can make her a co-host. Yes, yes, we understand the voice echo. I believe there's no voice echo anymore. Okay, I can see you, ma. So we also have, okay. I'm trying to give you access, ma. Have you joined the what's it called? The Google Meets?
Hello.
Yes, hello.
Yeah, hello.
Do you have something to say? You have something to say?
You just texted me you wanted to talk and you calls as well.
Okay, I'll call you back.
Nothing specific to it.
Sorry, I'll call you back.
On Google Meet.
Yes, ma. I'm trying to unmute you at the Zoom. All right. Sorry, give me a minute, ma.
Okay.
So we are having a little difficulties. Please. Apologies, please. We're still having some difficulties.
Can you hear me? Okay, I see comments, yes. Can you also hear me on Google Meet?
Definitely, ma'am. Likewise, yes.
So how does this work? I'll be speaking on, but it's kind of like I'll be hearing myself.
Oh,
Mhm.
okay. I think you have to kind of reduce the volume.
Okay, all right. So, but can you still hear me? All right, okay.
Okay, so thank you so much, Ma, for joining us. We really appreciate. Knowing that it's how can I put it now? So it's really an honor having you. We know that you're not in Nigeria, and that you really find the time to meet us. So we've been, trying all our possible best to have her. She's a very busy person, and it's really a great honor having her, with us. So, Dr. Temitope I don't know if I get that right. Fadikwe, right?
Yes.
So holds a PhD from University of Lagos, Nigeria, in cell biology and genetics, and specialized in the field of molecular genetics. So she's a chief research officer and head of the molecular biology laboratory at the Federal Institute of Industrial Research, Oshodi, that is in Lagos, Nigeria. So she was a visiting professor in the systems biotechnology group, Central Nacional de Biotechnology, in Madrid. So from November 2021 to April 2022, under the Science by Human program of the Foundation Majoras for Africa. So she's a recipient of several prestigious fellowship and grants since 2018. She coordinates and facilitates hands-on training molecular biology techniques workshop series at her home institute. Sorry, I think the thing is echoing. Hello? Okay, I think it's cool now. So she's passionate about research, developing STEM capacity in female scientists, and expanding molecular biology techniques, expertise among Nigerian life scientists. So she has really been a mentor to me personally. So being the CEO of GENOMICS Hub, so it's really a great privilege. Thank you so much, Ma. Welcome. Thank you for joining
You're
us.
welcome. Thank you for the invitation. So I have a little bit of a cough, but I'll just try my best to be as audible as possible. So if at any point in time I'm not very clear, then you can let me know so I can repeat myself. The topic I'm speaking on is, I think, more to me like a kind of generalized kind of intro to what we do when it comes to next generation sequencing. So I'll be sharing my screen now. I also sent the presentation to you. Can you see my screen, please? Can you see my screen? Okay, I just shared now. Can you see it now? Oh, I thought they couldn't see it. Okay, I'll reshare. Are we good now on Zoom? Okay, so the topic I was given to discuss with you all is next generation sequencing and its application in life science research. So To start, I would like to do a little bit of a reminder on the basis of everything that we do in sequencing is really all about, which is the DNA. So the DNA is a polymer that is made up of monomers that are called nucleotides, and then it joins one to another to form a very long chain. Each of those nucleotides is made up of a deoxyribose sugar, a phosphate group, and a nitrogenous base. And this is what the nucleotide structure looks like. The nitrogenous base, the deoxyribose sugar, and the phosphate group. Now, each of these nitrogenous bases are of two different types. We have the purines and the pyrimidines. The pyrimidines are there. We remember that each of these nucleotide monomers are connected by covalent bonds that are called phosphodiester bonds. And it connects the 5' end of one nucleotide to the 3' carbon of the next nucleotide. So as we see here in this reaction often leads to the release of the water molecule. So it's also called the dehydration synthesis. So now, the DNA molecule contains two strands, which are linked together by hydrogen bonds. These bonds generally appear weaker than the phosphodiester bonds, but because there's a lot of them, it stabilizes the molecule, and that's how we come about the double helix structure of the DNA. Now, the base